Good evening, Flatford Mill, and welcome to the BFB News. On today's news, exhaustion spreads through the students of Aberystwyth University and tension grows as Dane Lee continues to eat all the bacon at breakfast. But now, according to the optimal foraging theory, any food source that attracts an animal to feed from it will be abandoned when the GUD, or giving up density, is reached. This is the point at which the remaining food is not worth the risk it poses to the animal. Today I will be speaking to some students from Aberystwyth University who are investigating the effect of different environments on the GUDs of small mammals. So let's head up to the lab and see what they're doing now. So can you tell us what we're doing here?
We're going to be preparing the equipment for the giving up densities experiment that we're going to be doing. We're going to take the equipment out into the field where we can meet Ren Rose, who is going to show us the next step of the experiment.
Hi Ren, how about you show us what you're doing here?
Certainly, Cassie. What we're doing is placing each one of these 4 feeding stations along a 40-metre transect at 10 metres apart. This will hopefully entice the small mammals to come.
So Ren, what can you tell me about the habitats you've chosen?
Well, we've chosen 3 contrasting habitats in the hope that these will yield different giving up densities which we can then compare. The first is Valley Farm Hedgerow. The feeding stations that we laid are just on the other side of this hedge.
So this is your second habitat then?
Yes, this is Valley Farm Woods. It's broadleaf woodland and undergrowth, which should provide a lot more cover and less human disturbance than Valley Farm Hedgerow.
So last but not least, our final environment. What can you tell us about this one then?
Well, this habitat is almost riparian in nature, being on the edge of a large pond. It's broadleaf woodland, which will provide cover as before, but we expect a different range of animals to be present.
So here we are back in the lab with Laura to start analysing the results we collected today.
Welcome back, Cassie. We're going to start with the tray and then we have the tile and the hair tunnel to
Tell us what this piece of equipment's for.
Okay, we are going to put this in a box and use this UV light to look for any urine spots that are on the tile, and that way we can tell how many species have actually been in this area.
Delightful. And now we're going to look at the hair traps. So can you show us how you do this?
Okay, so we're going to cut this tape off, and hopefully on the inside of the tape we will find that there is hair from the animals that have passed through the tube.
What do you take from these results?
Okay, although the results though, there was no significant data, we can still take trends from the results that we did find. These show that the time of day was very significant in the foraging habits of the rodents, such as the night time there was lower risk of predation and therefore there was a lower giving up density. We also found that the location was very important in that covered areas are a lot more safer for the foraging rodents, and so for the GUD was also lower there.
One possible explanation for the non-significance of the difference between our day and night data is that the food may have mainly been visited by cathemeral species such as voles, which can be equally active during the day and the night. The explanation for the non-significant difference between our habitat data may be that optimal foraging patterns are strongly influenced by the distance from the edge of cover, which is actually pretty homogeneous in all 3 of our habitats because due to the patchiness of the habitats, none of them are very far from a path.
It could be an artificial presence that's scaring away our results, such as studies have found in relation to small mammal GUDs and artificial light.
Thank you to everybody involved in this fascinating project, but it's goodbye from me now. Stay classy, Flatford Mill.